gluten-free diet has previously been shown to reduce diabetes incidence in nut mice, which spontaneously develops type 1 diabetes. We hypothesized that this effect was mediated by the gut microbiota and that this effect could then be transferred to the pups of gluten-free fed nut mice. So in this study, gluten-free diet was given to pregnant NOD mice, or a gluten-containing control diet was given to pregnant nut mice, and this was given until the pups were born and they were weaned onto a normal gluten-containing diet. Interestingly, we saw that the diabetes incidence was decreased in the offspring of nut mice from the mothers on a gluten-free diet compared to the controls. And we also saw that the insulitis score and blood glucose levels were lower at 10 weeks of age. So a gluten-free diet has been associated with a shift in the gut microbiota, and to investigate this, we looked at their fecal gut microbiota by DDDE at 4 weeks of age when they were weaned, and we saw, you can see here the two groups, they cluster separately, which means that their gut microbiota was different. Later in life, you can see that they are all mixed together, so this means that there is no difference in the gut microbiota. So if there is a causal link to diabetes incidence, this seems to be mediated in the very first few weeks of life. Furthermore, we investigated the immune cell compartments by flow cytometry, as the gut microbiota, the vertically transferred shift in the gut microbiota, could be important in the early life priming of the immune system. So we saw here that the proportions of CD11b positive dendritic cells, they were decreased in the pancreatic lymph node, in the mesenteric lymph node, and in the spleen. And also the regulatory T cells were increased in the pancreatic lymph node. Here we also saw an increase in the CD8 plus T cells, especially those with a mucosal marker, which means most likely that these cells were primed in the gut and then migrated to the pancreatic lymph node. This was all seen at weaning, but later in life we could also see in the intestine that the IL-17 producing T cells were decreased in the offsprings of gluten-free fed mothers, both in the small intestine and in the large intestine. So now we want to further investigate what does the immune cell compartment look like in both the intestine but also in the pancreas, to see if we can find a link between these two. But for now, the conclusion is that the diabetes incidence is dramatically influenced by the diet that the mothers are fed during pregnancy and in the pre-weaning period. This is most likely immunological and mediated by the gut microbiota.